Good morning, students. So today we'll be studying about digestion and absorption of proteins in this class. The overview of this class will be what are proteins, digestion of proteins in different organs of your GI tract, that is in mouth, stomach and small intestine, and how they are absorbed through different mechanisms like active transport, facilitative transport and endocytosis, and the abnormalities of the protein digestion and absorption. So coming to what are proteins. So we all know proteins are made up of amino acids, and the amino acids are the building blocks of your proteins. So these proteins are the primary constituents of our body, and they are very essential for our structural and even functional well-being. So around 70 to 100 grams of protein is required per day for an adult man. Whatever proteins are subjected to digestion and absorption are obtained by two different sources in our body. One is our dietary source, that is around 70 to 100 grams per day. Another source will be the endogenous source, which is around 30 to 100 grams per day. This endogenous source is derived from your digestive enzymes and some of the worn out cells of the digestive tract. So this is how there are two different sources of proteins which are subjected to digestion in our GI tract or in our body. So these are the different sources or different foodstuffs from where we obtain proteins through diet. Okay. Coming to digestion of proteins. The major sites or the principal sites of our protein digestion in our GI tract will be the stomach and small intestine. And coming to mouth, there will be no digestion takes place in the mouth, as there is no any proteolytic enzymes or protein digestive enzymes present in our mouth and saliva. So that is the reason there will be no protein digestion takes place in mouth. Next, coming to digestion in stomach. In stomach, once this food from your mouth enters into the stomach, the bolus enters into the stomach, the food will stimulate the secretion of hormone in in the stomach called gastrin. So the hormone will be secreted from your stomach cells. So this gastrin is an hormone which in turn stimulates the release of gastric juice. Okay, so this gastric juice contains HCl, that is your hydrochloric acid, and some of the enzymes like pepsinogen and renin in infant. So pepsinogen is a proenzyme, or it is an inactive form of your enzyme pepsin, and renin is an enzyme which is present only in infants. So the pH of your gastric juice will be highly acidic, that is around 1.5 to 2.5. So we'll see another two other hormones which are secreted in intestine, that is secretin and the cholecystokinin. So these are the different hormones. Gastrin, secretin and cholecystokinin are the hormones which will aid the digestion of proteins. So coming to importance are the important function of your acid, the hydrochloric acid present in the gastric juice. This mainly secreted by parietal cells of your gastric gland. So this acid will provide acidic environment for the action of your pepsin. Pepsin is an enzyme which require acidic pH for its action. So that is provided by your HCl. The optimum pH will be around 1 to 2. So the denaturation of proteins is again done by your hydrochloric acid or HCl, that is called acidic denaturation of protein. So this acidic denaturation or denaturation of proteins make the proteins more accessible for their subsequent hydrolysis by enzymes. So this is the importance of your HCl present in gastric juice. Coming to the action of pepsin. Pepsin is a enzyme which is secreted or produced from your serous cells of the stomach in its proenzyme form or inactive form, that is pepsinogen. So this pepsinogen is converted to active pepsin by two processes called as autocatalysis, and one more is by your gastric HCl. So once it is converted to its active form, it will start acting on your dietary proteins or polypeptides, and it converts that proteins and polypeptides into smaller peptides and few amino acids. So this pepsin mainly specifically cleaves the peptide bonds of protein involving some of the amino acids like aromatic amino acids and acidic amino acids. I mean to say, this pepsin mainly acts on the peptide bonds which are formed from these amino acids mainly, that is phenylalanine, tyrosine and tryptophan, and acidic amino acids like aspartic acid and glutamic acid. And coming to renin. The renin is also called as chymosin. So it is the important digestive enzyme present in infants, as their staple diet for infants will be only milk. So that milk protein is the casein, which is digested from your enzyme called as renin. So this renin converts that casein into paracasein, which will combine later with the calcium to form calcium paracaseinate. This calcium paracaseinate can be effectively digested by pepsin, the enzyme. So this is how the renin will convert the casein into paracasein. So this is only present in infants and it is absent in adults. Coming to digestion in small intestine. So once this acidic chyme, that is the stomach content, passes into your intestine, that is in small intestine, so the low pH of this acidic chyme triggers the intestinal cells to secrete hormones called secretin and cholecystokinin. This secretin stimulates the pancreas to secrete bicarbonate and to release the pancreatic juice into the small intestine. So the function of this bicarbonate or pancreatic juice is to neutralize the gastric HCl so that it can rise the pH from 2.7 to 7. 2.5 to 7. Coming to the another hormone called as cholecystokinin, which will stimulate the secretion of the pancreatic enzyme secretion. Those are your endopeptidases and exopeptidases, where their optimum pH will be near 7. So this is how the secretin and cholecystokinin stimulates the pancreatic gland to secrete the pancreatic juice and the enzymes from your pancreatic gland into the small intestine. Coming to the enzymes endopeptidases. These endopeptidases, endo means inside, right? So these endopeptidases mainly cleaves the internal peptide bonds of your polypeptide chains to form or to convert them into smaller peptides. So these endopeptidases are mainly secreted from your pancreas in their inactive form or in the proenzyme form. So they are trypsinogen, chymotrypsinogen and proelastase. So these will be converted into their active form, that is trypsin, chymotrypsin and elastase for their action. Coming to exopeptidases. So the exo means outside. So exopeptidases mainly cleave off one single amino acid at a time from either end. It may be at the carboxyl end or it may be at the amino terminal end. That is how they will start their action and cleave a single amino acid at a time. So these exopeptidases are again two different types. One is the carboxypeptidases and aminopeptidases. Carboxypeptidases will be secreted in the form of procarboxypeptidase and aminopeptidase secreted by your mucosal cells. So this carboxypeptidase will act on the carboxyl end of your polypeptide chain or your protein chain, and whereas aminopeptidase will cleave the amino terminal end of the protein chain. So coming to digestion of proteins by small intestinal enzymes. So the luminal surface of intestinal epithelial cells secrete the enzymes called aminopeptidases and dipeptidases. The aminopeptidase, as I said, it is a exopeptidase which will cleave the polypeptide chain or the peptide bonds at the amino terminal end of your amino acids one by one to produce free amino acids and the smaller peptides. And the action of this enzyme require the cofactor called as magnesium and manganese. So this is about the aminopeptidase. And coming to dipeptidases, these dipeptidases, as their name itself suggesting, they will act on only the dipeptides to cleave that dipeptide into two free amino acids. So this is about the action of your enzymes present in small intestine. So this is the overview of your activation of proteases. Proteases is nothing but your proteolytic enzymes or enzymes which will digest the proteins. So in the stomach, the pepsinogen is secreted. That is activated by your gastric HCl or the active pepsin itself, and they convert the peptides or polypeptides into smaller peptides and amino acids. And in intestine, the hormones called as CCK, that is cholecystokinin. CCK is nothing but cholecystokinin and secretin. They will stimulate the secretion of pancreatic enzymes and the pancreatic juice. So the pancreatic enzymes, we have exopeptidases and endopeptidases. The endopeptidases are your trypsinogen, chymotrypsinogen and proelastase, and exopeptidases are procarboxypeptidase. So this trypsinogen is first converted into its active form trypsin by the enzyme called as enteropeptidase. So this active trypsin can convert the other endo and exopeptidases into their active form, that is chymotrypsinogen into chymotrypsin, proelastase into elastase, procarboxypeptidase into carboxypeptidase. So this is how the inactive form of your enzymes will be converted into their active form or they are activated. Coming to the bond specificity and site of action of your enzyme. So this table shows where the enzymes will act or where they are present and on which type of bond or which type of bond will be cleaved by these enzymes. The first one, pepsin, as I said, it is in stomach and it mainly cleaves the peptide bonds involved by these amino acids like your aromatic and acidic amino acids, that is tryptophan, phenyl and tyrosine, and acidic amino acids glutamic acid and aspartic acid. And coming to trypsin, that is in intestine, which is which cleaves the carboxyl groups are contributed by the lysine and arginine. Coming to chymotrypsin, it mainly acts on again aromatic amino acids, along with them leucine, methionine, asparagine and histidine, whereas elastase mainly acts on small non-polar amino acids, alanine, serine and glycine. And coming to carboxypeptidase, in intestine it cleaves the carboxy terminal end of the amino acids. I mean to say, it acts on the carboxy terminal end of the amino acids, and aminopeptidases, it acts on the amino terminal end of the amino of your polypeptide chain. So this is about your bond specificity, or the action of the amino acid, action of the enzymes on polypeptide chain. This is the overview of your protein digestion, where in stomach, in mouth, there will be no digestion at all. And coming to in stomach, so there the pepsinogen, that is converted to pepsin, and it converts, or it acts on the polypeptides, and it converts the polypeptides into peptides and amino acids. And once the chyme enters into the small intestine, the other small intestinal enzymes or pancreatic enzymes will act on the peptides and that converts into amino acids and oligopeptides, and these oligopeptides are acted upon by the enzymes called carboxypeptidases, aminopeptidases and dipeptidases to form free amino acids and dipeptides. Coming to the absorption of amino acids and dipeptides, so the transport of the amino acids at the brush border will be the absorption of most of the amino acids involves active transport requiring ATP, and they also need a specific transport protein. So the transport protein and ATP is required for the active transportation, that is active transport mechanism, which is energy consuming. The small intestine also possesses an efficient system to absorb some of the free amino acids, and the L amino acids are the one which are more rapidly absorbed by your active transport mechanism, whereas the D amino acids is absorbed through the simple diffusion method. So this is the mechanism of amino acid absorption. You can see here, 1 minute. So here is the active transport of your amino acids, that is along with the sodium, the amino acids are transported. Here it is by pinocytosis or endocytosis, where the large peptides or proteins will be absorbed through the mechanism called endocytosis. So these are the small peptides which are absorbed through your intestinal cells. So this is about your absorption of the peptides and amino acids. So we have one more cycle called as Meester cycle or gamma glutamyl cycle. This cycle mainly operates, this is again the amino acid transport system or the cycle, which mainly operates in the kidneys, intestine and brain. So here the glutathione is a tripeptide which will help in the absorption of amino acids. So this is an highly energy consuming cycle where the 3 ATPs are utilized for the transport of one single amino acid, and it's not a common transport system. Okay. So you should know the name of the cycle which is operating in the kidney, intestine and brain, and here the glutathione is the one which is biologically important peptide, and this is the peptide the tripeptide which will help in the absorption of amino acids. So this is that cycle which shows how the glutathione will transport the amino acid into the cells. Next, coming to absorption of intact protein. So the intact protein, mainly in infants and fetus, are able to absorb the intact protein through the pinocytosis. That is, example is your IgA, immunoglobulin A, which is absorbed intact from the colostrum of maternal milk to provide a passive immunity. Okay, so this intact protein, intact protein will not be absorbed by intestine, but some of the macromolecular absorption in certain individuals is the responsible for food allergy by forming an antibody, so that leads to food allergy in some individuals. Okay, so this is about the absorption. One is by active transport, another one is by endocytosis, another one is by your facilitative transport, and the intact protein by endocytosis mechanism, that is mainly in infants, which is for the absorption of IgA from the colostrum. So the genetic defect in the amino acid transport, they are 3 different disorders. One is Hartnupp's disease, another one is cystinuria and glycinuria. Here these defects are due to defect in the transport system. That is, the first one, Hartnupp's disease, is due to defect in the large neutral amino acid and small neutral amino acid transport, which includes alanine, serine and threonine, and isoleucine, leucine, valine, tyrosine, tryptophan and phenylalanine. So this defective system or defect in this transport system leads to disease called as Hartnupp's disease. And the cystinuria is the one more disease or a disorder which is due to defect in the transport system of basic amino acids with cysteine and acidic amino acids like glutamic acid, aspartic acid, arginine, lysine, ornithine and cysteine, whereas glycinuria is due to defect in the transport of glycine and amino acid. So this is about the defects or abnormalities of the protein absorption or amino acid absorption. One is Hartnupp's disease, another one is cystinuria and glycinuria. So the Hartnupp's disease is the one which is due to inability of the intestinal cells and renal cells to absorb neutral amino acids. In that, the tryptophan is the main amino acid. Defect in this absorption leads to the symptoms like pellagra, because the tryptophan is required for the synthesis of niacin, so whose coenzyme forms are NAD and NADP. Okay, so this is how the tryptophan absorption defect leads to symptoms of pellagra. So this is about your Hartnupp's disease. So the assignment questions will be digestion and absorption of proteins. So it will be asked as a 5-marker question, direct question, and list the proteolytic enzymes. How you are supposed to write? You are supposed to define them, what they are, and then make them into 2 different, that is exopeptidases and endopeptidases, and then write and Hartnupp's disease, which will be asked for 2-marker question. So what is the defect and why it is, and what are the symptoms, and you are supposed to write in your assignment book. So thank you. Any doubts, you can contact me.